glutamic acids. Then they go here, they go very special structure. This structure called methyl diamino pylimic acid. And then they go here, goes D-alanine, and they go another D-alanine. They connect like that. Now, by the way, what is alanine? What is this guy? Is this amino acids? It depends on the R. Is that right? The carboxy base and the amino acid base H is always depends on the R. What is alanine? Is that CH3? Okay. What is glutamic acid? CH2, then COOH. Is that right? That's glutamic acid. What is that guy? That guy is a little bit complicated. That guy is a little bit complicated. This guy is HC, NH2, COOH, then becomes CH2, we go CH, then go CH, NH2, then go COO3, something like that. Okay? So do you have to know about these? They connected with these. Now how they connected? Hydrolyzed. So we draw here, okay? This guy go. This guy gonna go. So we do H. We're gonna have a O. We have a C and a O. Okay? We have a, if we want to draw here C and O, then we go here is NH. Then we go here is C and H. And this is COOH. And then we go H. Okay? CH3. CH3 is right here. Sorry. CH3 right here. Then we go COOH. And this is the one we call the peptide bonds. And this guy is going to release, and then you add more and add more, like that. Add all these in. Okay? So that's become a cross link. Now, this is the one chain. If you have a multiple chain, what are you going to be? So it's very similar, looks like NAM, NAG. Then you go L-alanine. Then you go D, glutamic acids. And then you go this special guy, we just say methyl, okay? Then you go here, it's going to be the D-alanine. And then you go D-alanine. Okay, then you have another one like here, NAG, NAM, NAG. Now, of course, this is a long chain. Then you go opposite direction. Opposite direction, you have a L-alanine. Then you go here with D, glutamic acids. Then you go up with methyl. Then you go up with D-alanine. Then you go up with D-alanine. This is A. Now, how they connected? They need a glycine. Now, what is glycine, by the way? The simplest amino acids. And that is glycine. So, you can obviously see modified sugar, N-acetylmuramic acid, N-acetylglucosamine, with amino acid, like a chain or the cross bridge. So, how do you know a structure is cell wall? Go ahead to test the amino acids. If you see a numerous places, multiple places, is rich alanine, glutamine, and glycine, then you also do the chemistry. You find that muramic acid is there, possible it's a cell wall.
Is that right? That could be a quiz sometimes. They say there's a mixed bacteria material, then you do the chemical test, you find that a rich glycine and alanine, most possible what the structure it is. It could be a cell wall. It could be a peptide glycan. So, this is well explained here, what it is looks like. Okay, that is you can understanding. If you know a little bit chemistry behind that, and you can understand it well. Now, by the way, these amino acids, you go ahead to review it. We will talk in the examination 3 when we talk about the protein synthesis. We will mention that again. Okay. Next couple of slides is simple. We already talked about gram-positive cell wall system. You have the cell membrane, fluid mosaic model there. Paris plasmic space. This is a space is usually the enzyme will be accumulated because if the nutrition goes in, they could be breakdown, become a small piece, the bacteria can take it. Okay? Go up top, teichoic acids, lipoteichoic acids. The function we don't know, but we know for gram-positive bacteria. There is a heavy area which is called peptidoglycan. How about the gram-negative bacteria? What's the difference? Can you see? There is a double outer cell membrane system. First of all, is outer membrane and inner membrane, or plasma membrane, we just say. Number 2, their peptidoglycan is very thin. And the periplasmic space is right there. Also, they are attached with each other. You can even not see the difference. Then there is some unknown protein, like protein, like lipoprotein. But more important, they cause all the problems. The endotoxin is lipopolysaccharides, is LPS. So here is the thing. When you see gram-positive bacteria and the gram-negative bacteria, and here's another picture to compare both of them, you will have a feeling right now, and we'll talk about that later on. This is the reason most of the antibiotics, they are more effective for gram-positive bacteria. Because when you have a peptidoglycan, it is easy to target. And the gram-negative bacteria is difficult to do. The only way what you can do is cell membrane. However, the human being has cell membrane. That is the reason Streptococcus pneumoniae, you have antibiotics. Is that right? Staphylococcus aureus, you have. Streptococcus pyogenes, group B Streptococcus, all those you have antibiotics. You can take it. Do you have any medicine can prevent E. coli 0157H7, Salmonella, Shigella? No. So the reason is gram-negative bacteria is kind of resistant to those antibiotics because of the structure. So you can see it because there's no place you can attack. And LPS can go everywhere. When they release into the body, the endotoxin will be going. So that is a major difference between these two cell wall. Now, of course, once we know the structure, most of the antibiotics will attacking these type of links right here, attacking right here, they could break down here. Those are the 3 major places you can attack. So that's why different antibiotics, their principles looks like different, just their target is different. Okay, so just let you know about this. This is a picture, another picture. If you don't know, you like these kind of like a cartoon picture, this is a really good one. You see compare the difference. Heavy peptidoglycan is very thin. You have these 2 layers, this is only 1 layer. And the lipopolysaccharides is right there. You have lipids A and O polysaccharides. The O polysaccharides can be an antigen. Therefore, you have E. coli 0157H7. H is flagella. O is O polysaccharides is an antigen. Lipids A, because they have a lipids A, that's why gram-negative bacteria have 10 to 15% of the lipids, and the gram-positive bacteria doesn't have, only have 5%. Where are they? Some people say teichoic acids, lipoteichoic acids has a little bit amount of the lipids. But they don't know the function yet. If you look at all the textbooks, they will tell you teichoic acids, the function is unknown. Okay? Next one, we will be talking really quick to finish these slides, is capsule. This is a picture showing you Klebsiella ammonia. Collapsible ammonia, we will mention, when you see this on a agar plates, like a cheese, like a milk, we call it a milk-coiled colony. The reason is a heavy capsule. The capsule composed by polysaccharides, visible underneath the light microscope, and we could do is using dark field, using negative stain or capsule stain to stain it. We just mentioned it. It is a very strong protective structure for bacteria because it's resistant to the phagocytosis. The phagocytosis, which means white blood cell, will form a phagosome to eat the bacteria, and then the bacteria will release the capsule to resist it. They will also help bacteria to survive in a desiccation environments, which means a very dry, lower humidity environments, and they also could be excluding viruses and detergents. If you have antimicrobials, you have a cleaning, sanitizing to attack it, they will exclude all those type of the chemicals. So that's why collapsible ammonia and all those things will survive very well. Collapsible ammonia will cause ammonia, but not a major issue. The major issue is infant diarrhea and urinary tract infection. What is the symptom for urinary tract infection? Is you have a difficult to pee. Is that right? The terminology called dysuria. So we will mention this late detail in the examination suite. Okay, so I'm going to end up here, and Thursday we want to talk about the ribosome and the following all the other stuff.